more than 100 participants. And we had also a great poster session. We have a nice program lined up for so again, I'm Jim Briggs from the University of Houston. Very happy to welcome you to the second day of this conference. And let me turn it over to Don, who's going to give a little bit of instruction about navigating the conference system.
Good morning, everybody, and welcome. I'm going to share my screen with you. So this is Brella. Hopefully you all find it pretty easy to use. Here's the home screen. If you want to click on people, you can click on here and suggest meetings with people. It'll be ranked in the order of categories you checked of interest. The schedule for today and tomorrow is right here on the left-hand side. You can click on a speaker, see their bio. The stream tab is going to be really important today. The talks will be through here. And we ask that if you have questions for the speakers, please enter them here in the chat box. The conveners will be looking here for questions and ask them to the speakers. And the speakers will be coming over here after their talks and answer any unanswered questions. I'll hand it back over to you, Jim. Thanks.
And thank you, Don, very much. So it's my introduce the next speaker while she gets her presentation ready for display. So it's Serena Zanel, who's an MD, PhD. She was a Patronus Scholar who obtained first in physiology at the University of Cambridge before completing her medical degree in 2000. She's trained as a physician and also as a scientist, as you'll see, specializing in clinical genetics, which you'll hear some about today. She undertook a PhD at the Wellcome Sanger Institute in 2009, where she pioneered work in breast cancers through whole genome sequencing. And she's currently a Crook Advanced Clinical Scientist, an honorary consultant in clinical genetics at the University of Cambridge in the UK. And today, I see her presentation up. So today she will talk about harnessing the power of whole genome sequencing in analysis of mutagenesis in human cells. Pleasure to have you, Serena.
Thank you very much. Thanks for the invitation and for the very kind introduction. I hope you can all see my slides. So these are my disclosures. So today I was going to tell you a little bit about what we've been able to learn from whole genome sequencing in terms of mutagenesis, and how we then use all the mutations that we get from whole genome sequencing in a clinical way. I'm going to make a point sort of 3/4 of the way through that mutagenesis is actually rather normal, which is slightly at odds with how I was first trained in cancer genomics. And then I'm going to finish off with a clinical case study of how we use whole genome sequencing and mutagenesis in order to benefit patient outcomes. So the first bit is just a little bit of background. I do apologize if some of you know a lot of this already. But the value of having whole genome sequencing is that we can use all the data that's present in the cells that have turned into cancer cells. And of course, the cell will have started as being a normal cell before it's turned into a cancer cell. And the dogma in cancer research has always been hunt down those driver causative mutations that are believed to drive carcinogenesis. And for the longest time, it's been a case of about 1, 5, maybe 10 driver mutations per cancer. But in fact, a cancer's got thousands of mutations, not just the 1, 5, or 10 driver mutations. And there's thousands of mutations, which we've called passenger mutations, so initially just thought to be bystander events. But they're not actually random. They arise because of the different mutational processes that are going awry in the cell as the cell turns from being a normal cell to a cancer cell. Now, not everything is mutagenic, only some things are mutagenic. So this approach will only detect those biological processes that could cause cancer that do leave mutational imprints. So anyway, those mutational imprints, as I said, they're probably accumulating throughout life. And there are some things that are occurring right now as you're sitting here listening to me. So they're perfectly normal. So it could be errors of replication as an example. Some of us could be sunbed users or could be smokers, and we may damage DNA in certain exposed cells to these environmental mutagens. We could have diseases that cause bursts of mutagenic activity in our cells. Or you could have a DNA repair defect. Now, our cells are constantly under attack from its surroundings. And we have this sort of complex set of DNA repair pathways that are constantly mitigating this damage. But if you have a DNA repair defect, then that could leave its own characteristic imprint as well. So each of these mutational processes, we may leave a characteristic pattern that we call a mutational signature on the genome. And for the longest time, when you sequence a cancer genome, you get this sort of what looks like a random mess of mutations. But in fact, in the past, we've shown that if you had genome sequences from multiple tumors of the same tumor type, it's possible to use mathematical methods to extract mutational signatures. So by this, I mean, we try to recognize or identify the specific patterns for individual mutational processes. So here, the pink rectangles, and then we try to quantify them as well. So determine the size of those arrows. So to give you a sort of a crude example, if you're a heavy smoker, we should probably be able to detect that within the tumor, whereas if you're a lighter smoker, we'll see fewer mutations from tobacco smoke. Okay, so historically, we've presented signatures in this way. Historically, it was substitution, single base changes, simply because it was the easiest class of mutations to call in the early days of whole genome sequencing. But we presented this in this way, and really for biophysical reasons, that your neighbors mattered. So first we classify them according to the 6 main classes, C2A, C2G, C2T, T2A, T2C, T2G. But we take the immediate neighbors into account. So 5' we have ACGT, and 3' we have ACGT. So 4 by 4 is 16. And 16 multiplied by 6 gives you 96 channels in the whole pattern. So a signature is the whole 96 channel pattern. It may have some dominant features. In this case, the tallest bars are C2Ts at CPG. So 5' doesn't seem to matter, but 3' matters. So this is C2Ts occurring at CPGs, which is deamination of methyl cytosine, something that we know has been happening over the course of human evolution, and it's perfectly normal and happens in all our cells. So this is an example of an intrinsic or an endogenous mutational process that's happening all the time. And here's another example of, again, an intrinsic process, also deamination, but catalyzed by these enzymes called apobecs, and differing repair pathways acting on the damage from apobec can result in these 2 different signatures. So this is just to give you an example of some intrinsic signatures. There are also some signatures that are exogenous or environmental. And this is just to give you a feel of what they look like. The topmost panel is C2T mutations from UV light. This has been reported by others, by the way, well before whole genome sequencing. So this is very well known, well characterized. This is seen in malignant melanomas that have had UV exposure. This signature is due to tobacco smoke. Again, well described even before the days of whole genome sequencing, perhaps not quite as extensively, but the pattern has been reported before. And this one here is due to aristolochic acid, something that people ingest and identified in liver and kidney cancers. So you can really get, a sort of a record, an archaeological record of the exposures of an individual. So to give you a sense of what signatures look like in individual patients, I'm showing here 4 different cancer types: breast, cervix, this is a leukemia, and this is lung cancer. And each bar is a different patient, just to show you that every patient's got a number of different signatures, and they all have slightly different mutation rates. The arrow is pointing to the green signature 1, which is deamination of methyl cytosine, which I mentioned earlier. It's common, it's normal, and that's why it's present at a low rate in pretty much every single sample of every single tumor type, right? Because it's perfectly normal. And then you get things like signature 2 and 13 in pink, which is due to apobecs. Again, pretty common in really quite a lot of tissue types, but you will find that there are some cases that have a hypermutator phenotype. And I think this is an indicator of some abnormal biology here. My suspicion is replication stress, but you can get this strong hypermutator phenotype even from intrinsic or endogenous mutation processes. And then you can get signatures that are due to environmental exposures, as mentioned previously. So the arrow is pointing to the blue signature 4, which is caused by tobacco smoke. Note how it's only present in the lung cancers and none of the other ones. And note the mutation rate, which is really rather high compared to all the others. So the environmental mutagens tend to cause really large amounts of DNA damage and high amounts of mutagenesis. So that's just to give you sort of a whirlwind introduction to mutational signatures. it's a young field, it's still developing. I still learn every day from this area. The number of signatures isn't static. The types of signatures are not set, are not finalized either. I've talked about substitutions, but there are all sorts of types of mutations and therefore all sorts of other signatures from other classes. And there are mathematical issues, as always there will be when you're dealing with trying to model biology with mathematics. And factorizing a complex data set poses that problem of having multiple potential solutions. And the absolutely true number of signatures within a data set can sometimes be a little bit difficult to resolve. Another point to make is that the signatures don't all behave in the same way. And it's something that we all sort of assume when we're doing the extractions and the assignments of signatures to samples, because some signatures are more easy to identify. They have prominent peaks and therefore they're easier to extract from a complex data set. Some signatures are a bit flat and a bit harder to discern. So this is a PCA plot of attempts to extract signatures from a data set. All I want you to take away is that there are some like this. Signature 17 has got very distinctive features. And no matter what data set you have and however many times you do it, it forms a lovely distinctive cluster because it's very easy to extract. By contrast, you get some signatures like this. These are all different signatures forming this kind of crazy ring because they are a little bit difficult to separate from each other. So they're not as discretized. And that's important because some of these signatures are starting to be used for clinical classification of patients. So this arrow is pointing to signature 3, first identified in BRCA1 and BRCA2 deficient cancers, which have this BRCAness. Signature 3 is frequently used to identify tumors with BRCAness because tumors with BRCAness are believed to be sensitive to certain chemotherapeutic agents, specifically platinums and PARP inhibitors. But as plot highlights, it's difficult to be sure that you do indeed have signature 3 in your data set. And so stratifying your tumors based on the signature can be a little bit prone to erroneous, you can get erroneous classification. So I've pointed that out to you. I will also want to say that, while I'd love the signatures to be used clinically, I do want it to be used correctly. And one of the things that people have said to me is, whole genome sequencing is all very well and good, but it's just so vast. The amount of data and information you get per patient is just huge. How do you make sense of any of it? So no two patients have the same set of mutations. So I'd like to show you that even though no two patients have the same set of mutations, you can make some sense out of it when you look at all the data together. So I'm going to contrast whole genome sequencing data of 2 breast cancer patients here. All of the data is going to be here for you to see that there are distinguishing characteristics of these tumors for yourselves. So on the left, you have a 47-year-old woman with something called an ER-positive breast cancer. They tend to be cured quite well. She has got, these dots are all substitutions, and she's got 2,000 of them. She's got mainly signatures 1 and 5, which are kind of normal signatures. She's got, contrast that to the lady on the right-hand side. The lady on the right has got an inherited BRCA1 mutation, so she's got a DNA repair defect. And you can see that her whole genome plot, instantly you can see that it looks rather different. She's got many more dots around, so she's got a lot of substitutions, and the signatures 3 and 8, which I've mentioned earlier. Signature 3 is that BRCAness signature. They've also got different indel patterns. So this is the one on the left is kind of a classic indel pattern of relatively boring tumors, and the one on the right with this big bar here, these are deletions with a specific pattern of microhomology at the junction. And the tumor on the right has got all these green lines, which are tandem duplications. These are structural variants, so breaks in the sugar phosphate backbone, copying again immediately next door to it and reinsertion. These are tandem duplications happening throughout the genome. This is chromosome 1, 2, 3, all the way around to X and Y. So vast numbers of rearrangement beautifully dispersed through the genome. The last thing to note is that the patient on the right also has these pink lines here, which are copy number losses occurring throughout the genome. So that's 4 main classes: subs, indels, rearrangements, and copy number. Very, very distinctive on the right-hand side compared to the left-hand side. We don't have the time to do it, but if I could show you all the other DNA repair pathways in some of the other tumors, they really do have very classical appearances. Right, so given that we have all this information, can we make it easier? Can we design algorithms to just classify the tumors and spit us back a number for a probability of whether a tumor has BRCAness as an example, because BRCAness is something that all the pharmaceutical companies are hot on because they've got drugs for it. So We've applied machine learning methods on the mutational signatures, and we have created this algorithm. It's called HR Detect. It's based on multiple signatures of subs, indels, rearrangements, and copy number. It takes all the signature information in. It's a weighted lasso logistic regression model. We've trained it on patients that have germline mutations in BRCA1 and BRCA2 versus breast cancers that we know don't have it. So we've identified the features. We've got the weights. HR Detect is now going to be used on other cancers to see whether we can truly identify other tumors that might have BRCA-ness. So HR Detect scores are here. A high score predicts BRCA-ness and a low score predicts not. And this was applied on 560 whole genome sequence breast cancers. And as you can see, really quite a lot of patients actually met the cutoff of 124 or 560 to be precise. That's 22% of breast cancers. That's 1 in 5 tumors. Not 1% or 5%, which is where we thought we would be. It was 1 in 5 tumors. So that is potentially a whole bunch of people who could have a particular drug which they're not currently getting because people don't look for these mutational signatures. Right, so maybe we've got it wrong. Let's try to understand this thing on the right-hand side here. Let's look at it in more detail. So of the 124, we had our positive controls. We had 22 women who we knew had BRCA1 and BRCA2 mutations, and sure enough, we detected all of them. We didn't miss any of them. So how about all the rest? Well, we identified another 33 women who had familial BRCA1 and BRCA2 who didn't know about it before. So we opened a can of worms there. Informed a whole load of families that they could have BRCA1 and BRCA2 screening. We found 22 women who had acquired BRCA1 and BRCA2 defects in their breast tissue. So they were not born with a germline defect, but they acquired it sometime relatively early in breast cancer development. And in about a third of the cohort, we could not find a genetic or an epigenetic cause to explain the patterns that we were seeing. So we didn't just look for BRCA1 and BRCA2. We looked at all the other genes that are involved in the repair pathway, in the same repair pathway, homologous recombination, HR. And we just didn't find any other genes that could have explained why this third of the cohort also had this BRCA-ness pattern in them. So that's fine. We now have an algorithm. We know we can predict BRCA-ness. In fact, we can find additional patients that we didn't know had some of these BRCA defects. But actually, does it do anything clinically? And to do that, you've got to test it out in a population or in a clinical trial. So I worked with, had the good fortune to collaborate with Åkerborg and UN staff from Sweden. Northern Europeans, they have fantastic clinical and treatment information in spades. A lot of genomic studies in the past do not have this level of clear clinical and treatment information. And so we worked on a cohort of women who had, it was an area of unmet clinical need, triple negative breast cancers. Triple negative breast cancers, TNBCs, are, they're really the tumor type that tends to have pretty dire outcomes. It's a bit of a flip of a coin, about 50% of them don't do very well. And we can't tell which ones they are. The vast majority of other breast cancers tend to be cured, so it made sense for us to study this population. And I know that a lot of TNBCs are believed to have BRCA-ness. So here we've sequenced, whole genome sequenced, all TNBC patients in the whole of the south of Sweden. That's every district hospital in the whole of the south of Sweden without exception. It's a study called SCAN-D, and they've collected about 13,000 patients in total, but we of course couldn't afford to sequence that number of patients. We sequenced all the patients between 2010 and 2015. And what you're seeing here is that more than half of the patients had high HR Detect scores, so it's 58.6% had high scores. And that's probably what we kind of expected. Now, does it prognosticate in any way? So when we looked at how these patients, looked at their outcomes, we find that patients with high scores in black tended to do well, whereas the patients with low scores, this group here, tended to do badly. In fact, we had a small group of women who did not have any treatment at all because they were either too frail or they were too old. So they would represent the natural history of the disease. And this group here, in spite of getting treatment, they had the same kind of outcomes as the women who didn't have any treatment at all, which means whatever we're giving these women is not working at all. and we need to find better treatments for them. But critically, we can now identify the patients who are going to do well from the patients who are not. Okay, of this group that who are going to do well, some of you will say, well, you could have just sequenced the gene to find out whether they had BRCA1 and BRCA2 mutations. And I can tell you that we only found BRCA1 and BRCA2 mutations, and also a couple of others, PALB2, RAD51C. We had methylation and we had transcriptomics. We only found them in about 2/3 of the cohort. So again, in about 1/3 of the cohort, we just couldn't find a genetic or epigenetic driver. And so the question then becomes, if you cannot find the driver, can you still prognosticate on our outcome? And the answer is yes, you can. So this Kaplan-Meier plot at the bottom is basically that line, that black line, divided up into the patients where we can find the genetic defect in black and the ones that we can't in gray. And as you can see, there's really no difference between them. So taking the mutational signatures approach, you'd be able to identify the patients who would have responded well to treatment, something that which we couldn't do before. And you wouldn't necessarily be able to identify these patients if we had used, standard kind of just sequencing the gene, that targeted sequencing approach. Okay, so in fact, we've taken this another little step further, because currently all we can do is prognosticate. Does it predict outcome? Does it predict sensitivity to certain drugs? And of course, this is a very difficult area to try to crack because there's so much competition. But also it's kind of difficult to convince people to give PARP inhibition as a first-line treatment because a lot of clinical trials need to go against what's called standard of care. And standard of care tends to be rounds and rounds of horrible drugs. But anyway, we managed to work with Clovis and Nick Turner in London on a phase 2 window trial called RIO. So in this trial, patients are given first up, they're given PARP inhibitors, which is very unusual. They're given it as a window right at the start of treatment, because you want to see whether actually these patients are going to respond to PARP early rather than giving them rounds and rounds of other horrible drugs before they get the PARP. Okay, and then we're going to take samples from the patients at the start of their treatment and at the end of the 2-week window. So we've got samples at the start and at the end. We've also got blood samples because we want to know whether we can see any correlation with whether the tumor material is released in the blood. That's called circulating tumor DNA. And so it's, or liquid biopsy, we want to know whether there is any relationship. So this was a very small trial, it was just a proof of principle. And it was with a PARP inhibitor. And what we found first was that, again, more than half of the cases had high HR detect scores, and we found causes BRCA1, BRCA2, PALB2, RAD51C again in about 2/3, a bit more than 2/3. There was still a proportion of patients where we cannot find a genetic or epigenetic defect. The second thing we did was also to check that this correlated with functional deficits. And indeed, if you had a high HR detect score, those cases that had high HR detect scores had impaired RAD51 foci formation. When we looked at circulating tumor DNA dynamics measured at baseline and at the end of treatment 15 days later, we found that some tumors indeed showed a reduced ctDNA level following the PARP inhibitor treatment. And so the metric is called CDR15, which is the ratio of ctDNA copies, bit of amount of DNA at the end of treatment compared to at the start of treatment. So if you have a low CDR15, then it means that you've got activity against the drug. So we find that high HR detect scoring patients showed suppression of ctDNA levels at the end of treatment and low CDR15 indices, this group here. So in this study, we showed that HR detect high scores have a functional HR deficit, and this correlates with activity to this PARP inhibitor, rucaparib, in triple-negative breast cancers. And this kind of opens doors to exploring applications as a stratification tool in randomized control trials going forward. So I've explained at this point how we use mutational signatures, that there are signatures that are endogenous and exogenous, and then how we've used mathematical methods to design clinical algorithms, applied it to a prospective cohort, applied it to a clinical trial, small clinical trial. I want to also reinforce that, we don't just do analyses, we do some experimental work. I don't have enough time to go into all of this, but I will say that, we have worked with human isogenic models, and either CRISPR-Cas9 knocked out different DNA repair genes or treated the cells with different mutagens to generate a kind of a reference set of mutational signatures, generated under experimentally controlled conditions, because of course tumors are not experimentally controlled, right? We have no control on what these patients have been exposed to. And so we do have, a sort of compendia of all of these signatures. I don't have enough time to go into it. I just want to highlight that it's there. So this is a paper that's been published already, and this is a paper that's in bioRxiv, just about to go back to the editors in the next week or so. So this information is out there. What I would say is these are normal cells. These are not cancer cells. And the extent of mutagenesis is absolutely massive. And in fact, loads of other people have done now studies on healthy human tissue, and they showed that actually there's quite a lot of mutagenesis in our normal cells. And so really, what we need to do is to identify or distinguish the signatures that are actually clinically relevant from the ones that are not. I do also want to just sort of highlight the point that we've tried to explore here, which is that mutagenesis is not that abnormal. It is actually something that does happen in all our normal tissues. And so this sort of dogma, which is, and this is work I did with one of my colleagues in Cambridge, he's another PI, we sort of did this over coffee. It really questions this dogma that you had this classical way of thinking about DNA damage, of which there's different varieties, and that you had all these different DNA repair pathways, and they come and they mitigate everything and they keep everything as pristine as possible. But in fact, actually, maybe that's not what happens at all. That actually our normal tissues have a lot of somatic or acquired mutagenesis, including drivers. So drivers are happening in all our normal cells, thousands of passenger mutations, even little clonal outgrowths in all our normal tissues. It's only very occasionally that a clone becomes a dominant malignant clone and turns into a cancer. So how come so much mutagenesis, right? Are the DNA pathways not working properly, even in our normal cells? So we think that's unlikely. And to the best of our knowledge, the DNA repair pathways are actually operative. And we know that because the signatures you get from DNA repair defects are very marked. And so if our cells had DNA repairs pathways not working, you would see patterns. But we sort of hypothesized that perhaps our cells can be a little bit permissive to mutagenesis, and perhaps DNA repair is not allowed or restricted from being overzealous in its job. So this is purely hypothetical. If we had a DNA damage on the x-axis here, and wondered how the amount of DNA damage would affect the likelihood of cell division. So if the amount of DNA repair was unlimited, it would just keep going. Then if you had a huge amount of DNA damage, your cells would probably just die because it's trying to manage all this DNA repair because there's a cost. But if you had a degree of limitation to your DNA repair, then perhaps, it will just allow some mutagenesis to occur. It will just try to clean up what it can. And then, there's a bit of a trade-off that the cell prioritizes survival over genomic perfection. And that's probably why you do have quite a bit of mutagenesis in our cells. So we did perform some mathematical modeling. I'm not sure if this is going to work, but I'm going to try to explore what would happen if you had cells that had unlimited DNA repair. So here in blue, unlimited DNA repair, but the cells that can still do a repair but have some restraints are in cyan or turquoise. So in a situation where DNA damaging events are not really that deleterious, like in our cells, then most of the mutations are passenger mutations. During periods of high damage, the blue cells that have unlimited repair see all the damage and incur really high costs for repairing all that damage to the point of becoming depleted because it's just working so hard and using so much resource. By contrast, the cyan cells that do have limits on the amount of repair will only repair what's necessary in an effort to try to survive the onslaught. And so that's why you find that the cyan cells can keep persisting. Now, the thing we have to remember is that 98% of the genome is intronic and intergenic. The majority of mutations that arise as a result of DNA damage is unlikely to affect protein function. And so it's something that can be afforded by the cells, as it were. And we think that this is a reason why we actually get so much mutagenesis even in our normal cells. So I'm just going to finish off in my last couple of minutes with a case study. That was a little bit of a digression, that last bit, just to sort of reinforce the point about using all the mutational data in a whole genome. But actually, there's an element of interpretation. You have to read the whole genome, not just the driver or the signature independently. You have to put it all together and look at the germline and interpret the patient as well. And actually, in clinical medicine, we do this all the time. We never do one test on its own. It's informative in a context. And I hope that story comes across here. So this patient is 30 years old and he's actually known to me in the DNA repair clinic. He's got xeroderma pigmentosum type C. And he had his first basal cell carcinoma at the age of 3. XP is a gene involved in nucleotide excision repair. And so it's necessary to fix UV damage. And classically, the kids who have XP tend to have horrible sunburn, skin damage, and then they get skin cancers and die sort of in the second decade of life, at the age of 10, 11, 12 years old. These days, they get meticulous photoprotection. They do very well. They live to adulthood. But our patient here presented in January 2017 with this violaceous tumor on the medial aspect of his eyebrow. So it was diagnosed as an angiosarcoma, which is a rare tumor of the blood vessel lining. It's very rare. And he had wide local excision and all was fine. But by September that year, he presented with a lymph node met. By June 2018, he had liver and pleural mets. He had not responded at all to any of the angiosarcoma treatment, not one bit. By July 2018, this man was dying. He had extensive disease in his cranium, around his heart and lungs, in his liver, everywhere. And I was asked to look at his whole genome. So this is his whole genome. You can instantly pick up that he's got lots of red dots everywhere, very heavy mutagenesis. His drivers were very, very boring. These are, CDKN2A and P53 could be a breast cancer. He didn't have the kinds of drivers that are typically associated with angiosarcoma. And this is his mutational profile. And what I'd like you to spot is that this pattern here is very similar to skin cancer signature 7 caused by UV light. And so if you give this to most genomicists, they'll say, well, that looks like malignant melanoma. But in fact, you have to remember that this guy is an XP mutant. He cannot fix UV damage. So this level of UV mutagenesis is normal for him in all of his UV exposed tissues. This is not what's killing him. It is this that's what's killing him. So this is a pattern, a signature called signature 10, caused by proofreading mutations in polymerase epsilon. We struggled, but we did find the driver in polymerase epsilon. It was present at a subclonal fraction. So in the main bulk of the tumor, he's got a little subset of cells that we think is causing this signature and is what's killing the patient. And so we've got his UV signatures. We think the UV signatures has hit polymerase epsilon. It's a CPT mutation, and he's got this subclone that is driving his tumorigenesis. We had to do some staining. It was negative in the tumor, but remember, if it's in a subclone, then maybe when you do your staining, you get a false negative. So we repeated it in a metastasis and found high positivity for PD-L1. So polymerase epsilon tumors are believed to be sensitive to checkpoint inhibitor, and this staining was just to sort of help us, provide corroborative evidence to give him checkpoint inhibitors, something that is not licensed in the UK at the moment. Anyway, he did get the drug, and these were his scans before treatment. These were his scans 3 cycles in, and everything started melting away. By the end of 6, 7 cycles, everything had melted away. Today, it's 2 years later, and he's alive and well. And so that's a nice, happy story to end on. It's anecdotal, to be fair, but I, was trying to really communicate the fact that when you have whole genome sequencing data and all this mutagenic information, it's kind of handy to use all that information in order to interpret patients for treatment and interpret patient cancers for treatment. So that's me. That was my summary. I covered mutational signatures. I highlighted a clinical application, talked about permissive mutagenesis, and finished off with a clinical case study. This is my acknowledgement slide. A lot of fabulous collaborators really all over the world. And this is my team based in the University of Cambridge. And I'm very happy to take any questions. Thank you very much for listening.
Thank you so much for an amazing talk. Really fantastic examples of sort of computational analysis applied to, to the clinic and to the bedside, and I mean, rescuing somebody who was, pretty clearly, not going to make it, I think, without that kind of intervention. So I have 2 questions in the chat. One of those, first one is from Ilya. Can the mutational signatures for patients with BRCA-ness of unknown origin be analyzed to conjecture on molecular mechanisms? And also makes a comment that seems like rich biology is hiding in these patients.
I think, I think I think that's absolutely correct. I think what this has revealed is that there's so much information and there's so much knowledge there waiting for us to mine it. Certainly, we are working with the group in Sweden, and others are working on similar cohorts. They're very interested in that group of patients that look like they've got BRCA and we cannot find the genetic defect. I mean, there must be a cause. There have been one or two interesting findings. So, for example, we found that one patient had a retrotransposition insertion into the end of exon 1 of BRCA1. So we've turned off, or she has managed to turn off her BRCA1 gene in a way that we couldn't detect previously. So I think there's still ways of turning off genes that we don't fully understand biologically. Perhaps there's long-range ways as well. So I think there's a lot of that biology that hasn't been understood. And I'm sure there are more genes that can produce these patterns. So yeah, you're right. There's a lot of hidden biology there we don't fully understand.
And another question, this is from Rupa Thapar. So have you compared the HRD scores for the 4 different PARP inhibitors that have been US FDA approved? They have different PARP trapping effects. Do you think that would be reflected in the HRD score? And also, can you use your analysis to predict which PARP inhibitor might be the most useful for a particular patient?
Great question. No, I haven't done that exercise. I would love to do that exercise. I would have to get my hands on the 4 companies. Happy to sort of do that exercise, but certainly the companies are starting to get a lot more interested in doing this. You know, it's very interesting how a lot of the clinical trials have been very particular, right? So PARP inhibitors were designed initially for germline mutation carriers, but of course there's clearly plenty of other patients who have BRCA that are not germline mutation carriers, so it could have much wider application. But a lot of clinical trials that have been done, so you know, we've kind of gone to companies and said, let us sequence your tumors from these clinical trials. A lot of them haven't necessarily been done in sporadic tumors. A lot of them have been done in germline mutation carriers. A lot of PARP inhibitor trials have been done as maintenance PARP. So patients have had rounds and rounds of platinum treatment. We don't have a primary sample, so all we have is a metastatic sample at the end. And remember, it's just a snapshot. It's just an image of what has happened in the past. And if I do not have the primary sample, I cannot figure out whether the signature is now off. Now, in theory, if you have longitudinal sampling, then you'll be able to see whether actually your signature has come on or off. So, you know, the question is great. Ideally, we would like to be able to do this properly with all the right samples available. Did I answer all the questions? There were several parts to it. Can you also use your analysis to predict which PARP inhibitor would be useful for which patient? I'd love to be able to get to that point. We're not there yet for sure. And still, I think the main trouble has been that we don't have all the right samples available to us. And so I would really beseech people who are doing any studies and clinical trials, the power is in the samples and collecting the right samples. Please, please collect all the samples possible.
I had a real quick question related to your Swedish study. So you mentioned that you sequenced all of the samples that you could afford to sequence, I think 2010 or 2015, as I understood it. I had 2 questions. One is, did you feel that your analysis was sort of still, quite statistically robust, even though you had, 5 years is not a small amount of time, but still you had sort of a limited, somewhat limited data that you could analyze. And also, did you see any sort of regional clustering or differences among different, parts of your Swedish samples?
Yeah, it's a very fair question, because you can always increase numbers in these studies, and actually, the p-value was just significant. I think if we had a large cohort, it would have been better. I can say that we've now done this in a couple of other studies, so we know that it is robust. The trend has been seen recurrently. So yeah, you're right. I think ideally we would have liked to have had more samples, longer period, a more diverse population as well. This was south of Sweden, which was a very particular kind of population, so not really that applicable to quite a lot of the rest of the world. Yeah, so, you know, of course, we definitely need bigger numbers. I think there was no regional clustering within the south of Sweden. A question I get asked sometimes is whether there are ethnic differences in HR detect scores or high HR detect scoring cases in Asia versus Africa versus the West. And for sure, you know, we get more triple negative breast cancers amongst the Asian population and amongst African American populations, depending on the study that's being used. But certainly, of the samples we've seen, there are more triple negative breast cancers in general. But from within the triple negative breast cancers in those different ethnic cohorts, the behaviors of the signatures seem very similar. The signatures are not different. But the sort of the frequencies may be a little bit higher in certain populations over others. But within Sweden itself, within that area of south of Sweden, we didn't see any regional clustering. Maybe the numbers are too small though.
Well, thank you so much, and we appreciate you joining us. I know you're in quite a different time zone than most of us, and so we truly appreciate that. And so with that, we're gonna take a break and start back at 10 Houston time. And so thank you very much for a nice session, morning session, and we'll see you soon. Thank you.
Okay, everyone. My name is John Pim. I'm from Baylor College of Medicine. It's my great honor to host this session. This is a particularly interesting session because, as many of us know, that the field of biophysics has eyewitnessed two major transformation or revolution in the last few years. One, the cryo-EM as a method for determining protein structures in a very short period of time, wipe out almost the traditional field X-ray crystallography. And only a few weeks ago, probably, that the computational methods, I don't know if it wipe out the experimental approach, but it's pretty soon. I think by now, almost every computational biophysics in the world is thinking what's to do next. And I think the day that every experimental structural biologists will be thinking what to do next, that day will be coming soon. And we have two major speakers in this session. The first one is Matthew Baker, that I've known him for many, many years. And he was one of the first ones that directly modeling the protein structures against the EM density map without using template, for example. And even at the date, not that many people think the cryo-EM could solve atomic structure, but time has changed, and the great work he has done in the past now really has paid off, and I'm sure he has done something much better. So let's first hear Matthew. Matthew.
Thank you for the introduction. Yeah, so I'll start sharing my presentation here. So yeah, thank you for the introduction, and thank you for inviting me to be a part of this conference. And so I am going to talk a little bit about some of our developments in modeling protein structure directly from cryo-EM density maps. So probably by now, everybody has heard something about cryo-EM. You know, it's cryo-EM. I don't think it's replaced crystallography yet, but it's definitely made a huge progress in solving structures of large macromolecular complexes. Some of the highest resolution structures now in cryo-EM are approaching 1 angstrom resolution. And of course, we're looking at these complexes in nearly native environments. And I'm sure everybody probably also knows, you know, it was the Nature Method of the Year in 2015. The Nobel Prize in Chemistry was awarded to 3 pioneers in cryo-EM in 2017. So, you know, cryo-EM has come a long way in the last couple decades. But I do want to go back a little bit, kind of in history, and kind of give some perspective of to give perspective of where we're at now, and look at some of the kind of the early computational modeling that we did some time ago. So, you know, back around 2000, we were able to generate kind of the first sub-nanometer resolution structures, and people still call this mainly blobology, right? We were still looking at kind of, we could see these large complexes, but they were still kind of blobby in characteristics. But we were able to actually develop some computational tools to start analyzing these density maps. We're able to pull out secondary structure elements like alpha helices and beta sheets. And we're able to dock in high resolution structures with considerable accuracy using some additional tools as well. But this is typically what you might experience when you would look at a cryo-EM paper back in the kind of the early 2000s, where you would see a kind of a protein subunit pulled out with maybe some secondary structure elements or an X-ray structure fit into it. But even then, with this information, we could still essentially build some of these kind of low resolution cartoon models. And in fact, in our 6.8 angstrom structure of the rice dwarf virus, we used our secondary structures that we observed in the density map and the secondary structure elements that we could see in or predicted in the sequence. And we tried to match those based on kind of relative location and connectivity to produce some of these cartoon models. And while certainly these weren't atomic models, they gave us some basic idea of what the protein fold looked like. And of course, that led us to try and develop new methodologies that would actually get beyond these cartoonish models to actually produce real kind of atomistic models. And so we started work with various collaborators, including David Baker, that you'll hear in just a little bit, but using cryo-EM density maps as a way of essentially augmenting or helping to improve ab initio or even comparative modeling. So here the density maps could serve as constraints for folding as well as a method for selecting native structures. But of course, we really wanted to be able to build atomistic models directly from the density map. And around 2007, there were 4 structures that finally kind of hit that near atomic resolution range, where we could actually see, the details of the protein fold. So we could not only see secondary structure elements, but we could see the connections between them, and we might even be able to see some of the secondary structure, some of the side chains, particularly along the secondary structure elements. And so what I have up here on the right is a structure of GROEL that was solved to about 4 angstroms resolution and 1 subunit. And you can see that single subunit, it's still kind of not what you would expect from an atomic resolution structure, but there's definitely features there. You can see the alpha helices, you can see the beta sheets, even in places we could see the separation of the strands in the beta sheets, but you get an overall picture of what the fold of the protein would look like. So now if we went back to where we started a few years beforehand with these cartoon models, we decided to adapt the basic idea of matching the size and location of secondary structure elements that we observed in the sequence with those that we could predict, that we observed in the structure with those that we could predict within the sequence. And so we essentially use the same basic methodology to construct models for GROEL at 4.2 angstroms and bacteriophage epsilon 15 at about 4.5 angstroms resolution. These were simple C alpha models, and this was all kind of done ad hoc, where we were sat down and looked at the different sizes and the distances between the secondary structure elements and tried to generate all this by hand. So obviously, we wanted to develop a much more quantitative and computational approach to solving this problem. So just a year or two before all these kind of near atomic resolution structures came out, we had actually developed a method for identifying both alpha helices and beta sheets in the density map at the same time. This program SSE Hunter, which you can see an example of over here on the right, basically just gave kind of the geometric representation of where the secondary structure elements were. But one of the interesting parts about SSE Hunter was that actually we developed a novel density skeletonization feature, which actually provided us with kind of a topological map that related the different secondary structure elements. So no longer did we have this kind of simple cartoon, but we actually had kind of a real geometry that would allow us to kind of map the potential connections between the secondary structure elements. So here's just a simple example where we see a couple of alpha helices, and we can represent their relationship with a structure graph, basically just the size of the helix and the distance along the skeleton between all these helices. So we could also then now turn to our sequence, and we could construct a sequence graph, which basically just has the same information, a length and a connection between the neighboring secondary structure elements. And then we could use a simple graph matching approach to try and calculate essentially an optimal match of the sequence graph to the structure graph, which would produce a nice topological solution similar to what we generated by hand in the previous examples. And from this, we could then essentially just use our knowledge of protein biochemistry and essentially construct a model directly from the density map. And so this is a little bit more kind of codified into what we did, where essentially we identify the secondary structure elements, identify a topology, place the atoms along the skeleton, refine those atoms, and eventually get a nice model. And we were able to put this into our own kind of interactive protein modeling software called Gorgon. And it basically took all these kind of little bits of code that we had developed over about the last year or two and put them all together in a single interactive environment. So we can now build models directly from the density map using this kind of alpha helix beta sheet based approach. However, we started, it wasn't too long after that we started getting these kind of higher resolution structures where we began to see some side chain, what we thought was side chain density. We saw, the pitch of the alpha helix became much more pronounced, and we had these little protrusions where we would expect side chains to be. So we actually, in collaboration again with David Baker's group, developed a method essentially to take our simple C alpha models and these new higher resolution density maps and kind of turn those into full atom models and refine those models against the density through this kind of rebuilding process. And so this worked really well. And we ended up adapting kind of that approach along with kind of the standard X-ray crystallography approach with our ab initio model building techniques to produce kind of this final flow chart of how we construct full atom models directly from the density map. And so this is kind of where we stood for a few years, and it was successful in a number of cases. But there were a number of kind of caveats that we had when we were building these types of models. The successful de novo modeling really required that you had a couple well-defined helices that varied in length and position. The density skeleton could very easily be influenced by kind of gradients in the density, some the local variations in the densities, or path breaks in the density. And of course, some of the low resolution features, the density skeleton would sometimes get wrong. It often mistake, if you had a really low resolution alpha helix, it often ended up looking like a two-stranded beta sheet. So there were a number of caveats, and it really meant some of these little proteins that we had started to find in some of the reconstructions, here is GP10 from epsilon 15, made it very difficult because we didn't really have any secondary structure anchors to really build these models accurately. So we set off to build a completely new approach to constructing models directly from the density map. And so we really wanted to eliminate all of these kind of different restrictions. So we didn't want to use a structural template, obviously. We didn't want to use we didn't want it to be necessary to have secondary structure elements to create this kind of sequence to structure correspondence. We actually really didn't want to even use any explicit sequence information. Yet we still wanted the models that we got out to have some sort of, semblance to what a protein looked like. So we wanted to have some biophysical constraints. We wanted it automated. So, this is a pretty big list of, wants going from kind of these near atomic resolution density maps. But at the end of the day, this is kind of what we came up with. We wanted to be able to input our density map, kind of the expected number of amino acids in our protein, and a threshold for the density map, that we kind of believe represented the entire protein. So luckily at a GCC event, I had heard about this problem, the traveling salesman problem, which, it seemed like a very interesting route optimization problem, but it's basically, how does a salesman, visit a number of cities in the kind of the most efficient way? And so this is also, I guess, a kind of seminal problem for optimization, and it really represents kind of an interesting approach. But how does this really relate to cryo-EM density modeling? And if you'll hang on just for a second, I'll kind of go through a little bit, and you'll see the connections pretty clear. So here's a kind of a really reduced example. Let's say you have 4 cities that you want to visit. The traveling salesman problem looks at all the potential connectivities, calculates the distance, and then tries to find the optimal distance and the optimal path. So if you were going to visit Seattle, San Francisco, Los Angeles, and Houston, this is probably the optimal route. And of course, this is pretty easy when you think about just 4 cities, but all of a sudden, what if you have 4,000 cities or even 40,000 cities? So it's an NP hard problem, but there are a number of heuristic solvers out there that work reasonably well and are relatively quick for a smaller number of cities. So again, how does this relate to our cryo-EM density map? And I just put a kind of a blobby density map up here on the side. Well, it turns out if we actually think about our density map slightly differently, what if we take our density map and just represent it as a set of points? So you can use any type of kind of clustering methodology here, but essentially these points actually represent kind of the cities in the traveling salesman problem. And so we can then just essentially use the traveling salesman problem to kind of help us connect these cities. And while a protein isn't necessarily visiting cities in a map, what we can do though is we can essentially modify the traveling salesman problem to calculate kind of a minimal error distance. We know that the distance between neighboring C alpha atoms should be about 3.8 angstroms. So if we assume that the optimal connection between the optimal distance between the cities is 3.8 angstroms, we can use the TSP solver essentially to calculate the optimal path through the density map. So this is kind of what our original path walking procedure looked like. We gave it a density map. We kind of filtered it a little bit to remove some of the noise, generate some pseudoatoms, and then trace the path using Pathwalker. Well, as you can see, the path is it's roughly accurate, but you can see like the secondary structure elements aren't really represented very well here. So we ended up essentially developing a couple other routines that actually went in from this path trace, found the secondary structure elements, refined those, fixed those, reseeded the map, and computed a new path. And so this process we could iterate until we optimized our model. And once we had a nice model, again, these are all kind of C alpha models, we could then just basically go back in, convert our C alpha model to a full atom model, and refine it against the density map to produce our final model. So we first debuted our path walking approach back in 2010 at the 2010 Cryo-EM Modeling Challenge in Hawaii. And at the time, we were the only de novo modeling tool available. About 5 years later, there was another modeling challenge. And this time, there were several more competitors in the de novo modeling category for cryo-EM density maps. And our path walking tools had evolved, and we were now able to do quite a few more things than the original path walking setup. But we ended up finishing in the top category for de novo modeling in 4 of the 5 data sets we entered. So we felt very good with our path walking utility. And then in 2019, there was another model metrics challenge where we were given these 2 structures over here on the right, human apoferritin and a horse liver alcohol dehydrogenase. And basically, use your tool. We were told to use your tools again. And this time there were a lot of different entries from a lot of different groups. But what I want to do is I want to kind of share with you real quickly to show you kind of the whole path walking procedure that we used on one of the examples. And I'm going to include in all these slides that I'm going to show you next, actually I'm going to show you exactly what we did. I'm going to give you the command that we used and show you everything just to show you how simple this procedure is. So here's our density map that we started with. We apply a really, kind of light low pass filter to kind of remove some of the little bits of spurious density. Here we built the full model with our path walking utility that's a part of UCSF's Chimera package. It's available as a plugin, but essentially we built this nice initial trace directly from the density map. We then went and assigned a sequence using one of our utilities shown here. This is just a simple command line utility where our original path walking trace doesn't really have any directionality to it or any sequence assigned to it. So this path refine tool assigns the sequence and does a small bit of refinement, hopefully lining up some bulky sidechain densities and things like that. But a real simple sequence assignment and directionality tool. We then we use Phoenix to take our C alpha model to a full atom model. Again, use Phoenix to generate or to refine the model. Then go in by hand. You might fix a few little things. You might see that Phoenix did really well in most places, but maybe there's a side chain that got that came out of the density, or maybe you think you can find a better fit to the density. So we use Coot for that. Then we go back in, and now we built one monomer. We need to build up the entire symmetry, the entire assembly. So we apply the symmetry for our model to the whole density map using Chimera. Run another round of refinement with Phoenix, and end up here with our final model. So pretty straightforward, simple procedure. These are just some of the, kind of showing you, how we did in terms of our modeling. So this whole entire procedure that I mentioned took about 1 hour to generate this model. And I've compared our model with the known structure, in this case 2FHA. And you can see that, in most places we do really well. The one place where we kind of got a little bit off was there was a little bit of a break in the density over kind of, you can see in the far right of the model where, the side chain just got and the main chain atoms were just a little out of phase with respect to the known structure. This is the other example. This is the alcohol liver dehydrogenase structure. And actually, where we ended up doing not so great, we actually fit the density much better than the X-ray structure did, except for at the very end terminus where the density was a little bit broken. So our model's a little bit off at the end terminus. But aside from that, we're talking about half angstrom RMSD for both these structures, and they were computed in less than an hour and a half each. So, what I just kind of walked you through and told you how easy Pathwalker was, but I kind of wanted to break down some of the points here where, these are, the different tools that we used kind of in the different times that were required. So even though Pathwalking is our central tool here, We used eMan, we used Chimera, we used Phoenix, we used Coot. We used 4 different software packages throughout to kind of go from a density map to our final atomistic model. And so, these are things that we kind of learned that there were some difficulties along the way. And so the modeling challenges for us were very helpful. We figured out we could generate, take all of the eMan related and Pathwalker related tools and kind of put them into a semi-automated version. So we also figured out that we could take that automated version and then essentially make a nice little GUI in Chimera for it as well. So we had some UI improvements. We also were able to improve some of our modeling tools. We were able to develop this kind of fragment assembly method that helped us refine the structure a little bit better. We can generate models for multiple subunits with some very small tweaks to the traveling salesman problem. But generally, the path walking utility worked very well. So I just told you now again, worked really well, but where are the deficiencies? If that was it, I probably wouldn't be talking a whole lot right now. But we have a lot of stuff left to do in Pathwalker. Pathwalking, again, 4 different utilities. So really, that meant there's not only are you using 4 different utilities, but there's a data management issue of having to make sure that all the different software packages work nicely together. They honored the same origin for the density map and the model. So there are a lot of issues with kind of just integration. And then of course, not every tool works 100% of the time. So we did have occasionally some bad paths in some of the models, and those were due to maybe some pseudo-atom issues. Maybe the TSP solver could be improved, or maybe we could have additional statistics to tell us where the models are good or bad. So again, I'm just going back here and kind of reiterating that this whole process of pathwalking was done through several different software utilities. And so our first goal was for this latest version of Pathwalker, was to completely refactor Pathwalker and provide an integrated all-atom modeling environment. So now you would only have to work within one software toolkit. And so Pathwalking is now a part of Phoenix, and it basically integrates everything from map filtration all the way to model refinement from the final Pathwalking model. And along the way, because we did all this refactoring, we've improved the Pathwalking's tracing by adding new TSP solvers, adding probabilistic models. We've added new statistical analysis and new methods for refinement. So one of the great things about moving to Phoenix integration was that we simplified all of our dependencies, and we could use the model managers and the map managers within Phoenix, the Phoenix software as well, which meant all of a sudden, we had a huge library of new tools that we could use within the Pathwalking code. One of the things that we also did was we also examined different ways of generating our pseudoatoms, our cities. And originally we had used this kind of a simple k-means clustering method, but we found that we could use a bunch of different methods. That some of them were faster, some of them were slower, but for the most part, for our Pathwalking utility, all these different methods gave us about the same pseudoatom placement. So the pseudoatom was pretty good, the placement was pretty good. But one of the things that we found was that this mean shift filtering technique for low resolution structures actually gave us a really good kind of path through the density map. In the low resolution structures, a lot of times the k-means or the GMM clustering tools would kind of put side chains in kind of these big bulky side chains, and with this mean shift clustering, we're able to kind of eliminate that in some of the low resolution structures. So that's now an option within Pathwalking. Another great advantage was that we switched from our kind of proprietary TSP solver to the Google's OR tools, which contain several different optimization tools, including their own TSP solver. Their TSP solver had a bunch of different advantages, including having multiple essentially routes or multiple starting points. You could start from different, there were start and stop points, there were different local search strategies, and a whole bunch of different routing options that really gave us the ability to kind of fine tune the TSP solver for de novo modeling. We also finally implemented another layer of statistics in path walking, where we were able to generate multiple models very quickly. So here, for our simple example, we essentially were able to generate 99 models in about 4 minutes and look at kind of statistically how these models were similar and different, and essentially assign each connection a probability. And so that now gives us a whole different way to observe how well our models are doing. And that also led us to kind of a new refinement tool where we could evaluate the path, look at the good parts of the path, and essentially reseed any of the bad positions along the path, and build better models. So here's just an example. This was our model challenge structure here in green. Our completely automated structure with Phoenix from the same from the path walking tools that I was just talking about, overlaid on the density map. And these structures are, about a half angstrom different in terms of RMSD. The biggest difference is our automated approach took about 20 minutes compared to a full hour for our model challenge approach. So with the last few minutes, what I want to do is real quickly just talk about one example, the mud crab reovirus. And this is a structure that we use all of our kind of older path walking tools on. But I just want to show you kind of the power of what path walking was able to do in combination with a kind of a near atomic resolution density map. So mud crab reovirus is a reovirus. It's got a 12 segment double-stranded RNA genome, and it's if you're familiar with reovirus, it's kind of a smoother capsid. It doesn't have a turret, and it's got 2 layers of a double layer kind of capsid shell. And so this is the reconstruction of mud crab at about 3 angstroms resolution. We actually did several different reconstructions of mud crab. This is just kind of the wild type quiescent version or quiescent reconstruction. We have an actively transcribing reconstruction as well at a slightly lower resolution. But anyway, you can see just kind of from the zoom in, you can see the features, and you can really begin to see, even at this kind of low resolution picture, that there are some really distinct features in the density map. So this is one asymmetric unit shown here, and it's very typical of reovirus. It's got 2 layers of proteins. The outer shell is made up of VP11 and VP12, and the inner shell is made up of primarily of VP3. We used our path walking tools to construct models for all these subunits. And so these are just the subunits here. The one subunit I didn't mention in the previous picture was actually the RNA-dependent RNA polymerase, VP1, but we were able to build that as well using path walking. Path walking, as I said, doesn't require any templates. So, that's very important in this case. VP11 was a completely novel structure. It has a completely novel fold. And interestingly, even though the mud crab reovirus belongs to a family of kind of these non-turreted reoviruses, the VP11 protein actually sits in the same kind of location as some of the clamps and other reoviruses. So while we don't know exactly what VP11 does, we have a very distinct clue based on its location relative to some of the other reoviruses. And taken all together, this is really interesting because it suggests a completely different evolutionary path for reoviruses. So this is the inner capsid protein VP3. And again, all these models were constructed directly from the density map using PathWalker, using a very similar protocol to what I mentioned earlier in the talk. VP3 actually forms kind of the pseudodimer on this inner layer. So there's an A subunit and a B subunit, and while they are the same protein, they actually have kind of they have very different conformations. Actually, about 7 angstrom RMSD between these two conformations. And not only is kind of this overall rotation different, one of the biggest differences is what happens at the N-terminal arm of VP3. In VP3B, the N-terminal arm actually stretches around the 5-fold vertices and interconnects 3 different VP3 molecules. However, in VP3A, the N-arm adopts 3 distinct conformations, and 2 of these conformations actually interact with the RNA-dependent RNA polymerase. The RNA-dependent RNA polymerase is actually offset from the 5-fold vertex, so only the 2 VP3A N-arms are actually interacting with the polymerase. The other 3 VP3A N-arms are kind of off, providing more capsid stability than interaction with the polymerase. So this is just kind of a zoom-in view of that real quick. This is the polymerase in gray, and then yellow and blue are the 2 copies of VP3A, and you can see how the N-arms come down and actually interdigitate the polymerase. In fact, in one of the VP3As, this kind of long stretch actually forms a nice strand that forms a larger sheet with a couple strands from the RNA-dependent RNA polymerase. And of course, I mentioned very briefly at the beginning about this case study on mud crab reoviruses, that we'd actually reconstructed this not only in the wild type kind of quiescent state, but actually an actively transcribing state. And what was interesting is that when we looked at the reconstruction, we could actually see double-stranded and single-stranded RNA entering or double-stranded RNA around the polymerase and single-stranded RNA entering the polymerase itself, as well as some nucleotides in like the priming location of the RNA-dependent RNA polymerase. And so this was all only possible because we were able to generate high-quality, accurate models very rapidly directly from the density map. Even though a lot of these models had some structural similarity,
Matt, Matt, you have 2 more minutes.
Yep. That was it. That was the last slide. I just want to say one last thing. The computational modeling now in cryo-EM can actually give us very high quality models. We don't need giant supercomputers. All this work was done on a simple, desktop computer, an iMac circa 2015. So, this is a very accessible technique now for a lot of people. And finally, just leave this slide up here. These are a bunch of people who worked on the project. Corey Rick most recently has been working on the refinements to path walking. And I've got a little video showing right now while I take a question or two of the actual model building process that we did for the model metrics workshop with our path walking GUI and Chimera. All right, thank you.
Thank you. Thank you, Matt. This is beautiful. And let me ask you first question, sir.
Yeah.
Don't you have to know the beginning point of the path in order to model the path?
Originally, we thought so. No, we don't. The way the traveling salesman problem works is basically it will determine the starting and the end point for you. And in most cases, the starting and end point in the density map actually are pretty clear. They're essentially dead ends, right? So if we look at kind of the distribution of pseudoatoms, they actually have the least amount of connections. So in most cases, we don't actually have to express the endpoints specifically. But that is an option that we can add, or that is available in path walking, that where you can specifically put in the beginning and end point.
Okay, so the question from audience is that why do you think it's helpful not to use a sequence, or where do you place dots in the path? You probably have an answer to that.
Yeah. So really the question became, why aren't we using sequence? Well, the problem was that at 4 angstroms resolution, how reliable are the blobs that we're seeing, right? Are they sidechain density or are they noise? And really, some of our early reconstructions, we weren't sure if what we were seeing were actually side chains or if they were noise. So we wanted to kind of be agnostic to that. We wanted to, just see if we could trace the overall protein fold and get the correct topology. And if we had that, then we felt like we could then go back in and essentially use kind of our biophysical constraints to improve that and give us a more protein-like feature that did have registration with side chains.
I noticed you do iterate. There's another question that what sort of a EM map are you using as a starting map? So I guess it's asking what kind of resolution you need to be able to generate a good result.
Yeah. So we've tested path walking from about 2 angstroms all the way out to about 6 angstroms. And obviously the results are what you would expect, the kind of the higher resolution, the better your model is going to be. But if you do have a protein that is primarily, alpha helical, going out past 5 angstroms as possible with path walking. But kind of the baseline that we generally say is if you can see a separation of strands in your beta sheet, path walking will probably work.
So I guess the next 2 questions, the one is asking, how can we use path walker? You said it's available in Phoenix now, right?
Yes, yeah. So originally, Pathwalker was part of the EMAN2 package, and actually it is still, if you download EMAN2, it's still part of that package. But in the last about 6 months, we've shifted all of our development over to Phoenix. So I guess probably the next major release will have all the pathwalking tools. Right now, it's only available as a command line. It's actually not part of the GUI. Corey should be working on the GUI, and hopefully we'll have that done before the next official release. The EMAN version does have a little widget that's available within that you can load into Chimera, and so you can use path walking within Chimera.
Sure. I think another question is that if there is a disorder region, can you actually not get lost? I guess.
Yes-ish. No, if there is still, if the map is disordered in particular regions, or if there's a break, that's where we see our highest amounts of error. And a lot of time what we see is when we do have kind of this large regions of error, we'll end up doing very well in bits of the structure that are well defined, and then have kind of higher errors in the disordered region. Pathwalker will build a model in the disordered regions. It will, if you tell it to put in 225 amino acids it will put in 225 amino acids. It won't necessarily necessarily distinguish between the good ones, or it didn't until recently distinguish between the good points and the bad points.
The last question, I think there was a continuation of a previous question. So what sort of a question is suitable for path walker? Don't mean the resolution. I don't exactly understand this question. I guess you can. This method applies to any kind of a protein, right?
Yeah. So
soluble, unsoluble, it doesn't matter, I guess.
Yeah, virus. So virus, membrane proteins. Basically anything in the resolution range. Again, path walking tools. If your map can be read by EMAN or by Phoenix, you're good to go.
Okay. All right, Matt. So glad to see that you have done so well. And we do have to go move ahead. And Matt, you can get into the bladder region to answer the remaining questions, if not picked up or answered clearly. And should we switch to the next speaker? Thank you, Matt.
Thank you.
Mm-hmm. So our next speaker, I guess, who doesn't need much of an introduction, Dave Baker, truly leading expert in the field of protein structure prediction. And many of us know that up to very recently, before that, everybody thought that what David has done is the roof for structure prediction. That's a very difficult to surpass. And all of a sudden, AlphaFold 2 or DeepMind folding method has done better. But I guess, without the pioneer work of generations of scientists, especially a scientist like David Baker, who has done truly amazing work in the structure prediction, even the AI methods, whatever the AI method, cannot achieve what it has done today. So it's a truly a pleasure to hear what David wants to say, because everyone in the world now, especially in the field of protein structure prediction, is thinking what to do next. David, are you there?
You're muted, David.
Can you hear me now?
Yes, David.
And
Great.
let's see. Do you see my?
Yeah, we do see PPT.
Am I in presenter mode? How about that?
No, you are now. Yes.
Okay. All right. Well, yeah. Thank you very much for the invitation to speak here, and I'll come back at the end if there's time to the impact of deep learning on protein design. But my talk is really going to be focused on protein structure prediction and design. My talk is really going to be focused on protein design rather than structure prediction. And what I want to tell you about is what's become now possible with de novo protein design. And the basic principle which underlies what the work I'm going to describe to you today is that proteins fold to their lowest free energy states. So in the structure prediction mode, that means you, given a sequence, you search for the lowest energy state. And if you want to instead design a brand new structure, you make up the backbone, you come up with the backbone for that structure, and then you have to find a sequence whose lowest energy state is that backbone. And we've developed a computer program called Rosetta that over the years for doing the energy calculations and the sampling of sequences and structures required to do these calculations. So what I'm going to focus on today is de novo protein design. And this is an interesting problem because sequence space is very, very large. So with 20 amino acids at each position, there are for 100 residue protein, there's 20 to the 100th possibilities. And this is a very, very huge number, 10 to the 130th. The number of naturally occurring proteins is much, much smaller. So if we depict all of sequence space here, it's the naturally occurring proteins and naturally occurring protein families occupy a very small amount of it. Now, most protein engineering to date has involved taking naturally occurring proteins and making small modifications. And what I'm going to tell you about today instead is de novo protein design, where we make up completely new proteins from scratch to solve, to try and address current day challenges. And I'll just mention that the space is actually underestimated. We've been doing a lot of work recently incorporating unnatural amino acids and even unnatural protein backbones into design calculations. So the actual space is in fact far larger than 10 to the 130th that we can explore with these methods. So I'm going to start with to illustrate what the methods can do with a description of what we've been doing since the COVID pandemic broke out in the area of developing COVID antivirals, diagnostics, and vaccines. So shortly after the genome sequence was released, we built models of the three-dimensional structure. And then once the EM and crystal structures were released, we switched to using those. And I have to say it was wonderful how quickly those were shared. So I'll start off with antivirals. So here what we sought to do is to use de novo protein design to design small proteins which bind to the coronavirus spike protein and block its interaction with the ACE2 receptor. And we used two approaches. In the first approach, we took a helix from ACE2, as shown here, and built small proteins around it, as illustrated here. This is the receptor binding domain of the spike protein. The second approach, we generated hundreds of thousands of small protein scaffolds on the computer, and then docked those against the RBD to identify those that were shape complementary to the to this portion of the surface. Now, in both cases, we designed this amino acids at the interface to make very low energy interactions with the RBD. And we got a number of hits from both methods. I'm going to show you one of the best ones. This is from the second approach. So this is the completely de novo approach. There's no information from ACE2 being used here. So it's a small 3-helix bundle. It's 55 amino acids. And you can see its shape is very similar, very complementary to that of the virus. And that's particularly obvious when the side chains are on there like that. It's a very stable protein. It doesn't melt at 95 degrees. Here is the cryo-EM structure of this protein bound to the spike. So here's the spike trimer, and here we see the little 55 residue protein bound looking down from the top. And if we superimpose on the, if we take one of these experimental structures here, and here's the RBD, and we superimpose on our design model based on the RBD, you can see that this protein is binding to the spike protein exactly where it was supposed to bind. The magenta and gray are almost identical. And when you look closer at the side chains, you can see that we've designed this with really very high accuracy. And by we, I mean Longjin Cao, who really is the hero of this story. So in collaboration with Brett Case at WashU in St. Louis, we looked at the ability of this protein to protect cells in culture. So this is the protein I just showed you in magenta here. Here, virus is being added to cells, and we're looking at the protection of those cells by the binder. This protein blocks infection with an IC50 of 15 picomolar, which is 0.15 nanogram per mil. That's in fact the most potent compound currently known to block viral entry, at least to our knowledge. The collaborators at Dan Bruce's group at Harvard, they administered this protein to hamsters through intranasal administration, and then infected them with virus 24 hours later. And the hamsters that got the sick, whereas those were that's two different versions in black and red did much better, and by 8 days or so were indistinguishable from animals that never saw the virus at all. And this is another experiment from our collaborators at Brett Case at WashU. This is now giving mice with the ACE2 mice this protein intranasally, and again, mice that do not get the binder get sick, and those that do are healthy. And in this case, they went ahead to look at the spread of the virus, and it was really shut down. So this is the amount of virus in different tissues from animals that got the binder, or did not get the binder. And you can see the animals that got the binder were pretty much completely protected. So this is kind of an interesting protein. I've just summarized what I've told you here, but I also told you it was very stable. And we can express it at very high levels in E. coli. So this is a whole cell extract. This is the design protein here. Remember, it's just 55 amino acids. So you want whole E. coli cells expressing the protein, you get this big band. If you boil the cells and then spin, a lot of the cell protein spins out, and what you have in the supernatant is pretty much just it's a reasonably pure preparation of this protein, which we can then clean up in one step on DAE. So we're very excited about this as a very low cost of goods and widely distributable prophylactic and therapeutic for the virus that could just be given through a spray. So we're kind of exploring the development of this. And one thing that we're very excited about doing is improving this design pipeline, so we could design small proteins like this to any new pandemic threat within a couple weeks of its emergence, because after all, everything's going on the computer. So the next thing I want to tell you about is diagnostics. And here we've been developing for a number of years switchable systems, which can have an off state and an on state. And in the on state, they are luminescent, whereas in the off state, they are dark. And so what we've done is to couple binding of analytes, things you want to detect, to the opening of the cage of the system thermodynamically. And so in this case, what we've done is to embed within the off state the binding protein I just told you against the virus. So when the virus is present, thermodynamically, this on state is open and we get luminescence. And you can see this here. So this is adding the spike trimer. You can see shortly after adding the spike trimer, we get a big increase in luminescence. In the absence of trimer, there is very little luminescence. And we can detect this on plate and on paper. We're currently working to increase the sensitivity of the system further by incorporating multiple binding domains so we can get very high sensitivity. And we can use the same approach to make sensors for a wide variety of compounds. Remember, this is a completely de novo design switch. So for example, troponin, botulinum toxin, antibodies against the virus. And this in these experiments, what we're doing is taking a large collection of sensors we've designed and adding to each of them a given target like troponin C, and we only get a response from the sensor which is designed for that. So they're quite specific. And the final thing I want to tell you about is vaccine design. And I should emphasize in all three cases, we've been working on the protein design methodology for several years before. For example, in the antiviral stuff we had really it was perfect timing because we had recently, Longjin and Brian Coventry had made a real advance in our ability to design binders to arbitrary protein surfaces. And so we basically, when the pandemic broke out, we can take everything we've been developing and apply it. So the vaccine work starts from methods we developed a number of years ago for designing self-assembling nanoparticles composed of two different components. And here's the basic idea. We can form these icosahedral structures. And when you look at them in EM, they are very similar to, they basically are very homogeneous particles, and they are very similar to the design models. We've solved crystal structures of a number of these, so we can design these self-assembling systems very accurately. My colleague Neil King, and everything I'm going to tell you about in the next two slides is work from his lab, is basically took these nanoparticles and put the receptor binding domain that I just told you about of the spike protein on their surface. And here's what these particles look like after that. And these have really remarkable effects on in animals, both in eliciting neutralizing antibodies and in protecting against infection. They're far better than just the spike trimer alone, which is what the basis of the current mRNA vaccines is. So these will go into clinical trials very soon, and we're hoping they can contribute to combating the pandemic. Okay, so I'm now going to leave coronavirus and tell you a little bit about some of the other sort of more recent design work from my group. And on the topic of nanocages, we realized that that we could use as building blocks in these nanocages, not just designed proteins, but we could actually use naturally occurring proteins that are interesting in themselves. And in particular, we realized we could use antibodies as a building block. And if you think about it, icosahedron has a 5-fold symmetry axis and a 2-fold symmetry axis. And we realized we could put the antibody on the 2-fold symmetry axis, provided that our design component had the right geometry to hold these antibodies in the right relative orientation to form an icosahedron. And this is work of Robbie Devine. This is comparing cryo-EM maps of these designs that I'm telling you about now with Robbie's design models, and you can see they're very close. Robbie, in fact, used this method to design antibody nanoparticles that have different symmetries and display different numbers of antibodies. So here, this is a dihedral one with antibodies sticking in opposite directions, which is very exciting for applications like BITEs, where you want to bring cells together. Here's a tetrahedral design with 6 copies of an antibody, an octahedral design with 12 copies, and then icosahedral ones like I just showed you with 30 copies. So these are interesting as delivery vehicles, but I'm not going to talk about that today. But we have been doing with these is exploring the generation of signaling pathway agonists by clustering receptors. And here's just one example. This is taking the octahedral design protein, mixing it with an antibody to the B cell protein CD40 to produce these cages. You just add the antibody alone to the cells, to B cells, there's no activation. But when it's wrapped up in these nanoparticles, you get strong activation. And the beauty of these proteins is you can take any off-the-shelf antibody and just add these proteins to drive the assembly of different nanoparticles. And we're using this now to make superagonists for a wide variety of pathways. So on this topic of self-assembly, I'd like to now talk about unbounded materials. And here I'm showing you 2 proteins, a D3 protein and a D2 protein, that means dihedral symmetry, which we've arranged in the plane in this manner to direct formation of a hexagonal lattice. And when we make these 2 proteins in cells, what we observe, and then take the pellet out, what we observe indeed is this hexagonal lattice. And if we fuse GFP near the corners of the lattice and then look, we see the little GFP here. Ariel Ben-Sasson and Matt Johnson then went on to purify the proteins from E. coli and purify them separately, and they can get them very pure at high concentration and then mix them together. And when they do that, these very regular and very, very large hexagonal arrays form, shown here, and they're very, very regular. And if you take this image and you average, you can see the density for the 2 design components. One is in magenta and one is in blue. So they're very close to each other. Now, I don't have time to show you, but this is enabling us to do some really interesting cell biology experiments. First of all, we can cluster receptors much more extensively than before by putting receptor domains on, binding domains on these. And second of all, and what we find is that when we add these arrays, we can really shut down endocytosis because the receptors cannot recycle against this force at the membrane. It's been a great collaboration with Emmanuel de Rivry, who's done some really beautiful quantitative microscopy to characterize the formation of these on living cells. So it's sort of the beginning of a new sort of design synthetic biology where we can design materials that interact with cells in different ways. So the next thing I want to tell you about is the problem of identifying specific subsets of cells in a population, in the body, for example. And this comes up because often the cells you want to detect are not distinguishable based on just one surface marker. So in cancer, for example, some cancers have a single unique marker you can target, but many don't differ from naturally occurring cells in a single marker, but differ in combinations of markers. So how would you target cells that have 2 markers, but don't have either marker alone? And actually, maybe the idea is more clear. So we used a system we've developed, which is related to what I described for the diagnostic, where we have a cage and a key. And the cage has a recruitment motif in this case, but it's sequestered and cannot interact with the effector, which I'll show you in a moment is a CAR T cell, until the key comes in. And so cells that have both markers get the key in the cage and the effector is present, is exposed. But if they just get the cage alone, it's closed. And if they get the key alone, there's no recruitment motif. So only in this case is the effector exposed. And let's just focus attention on this last column here. So in this case, we have 2 tumor cell markers, which are HER2 and EGFR. And then we have a healthy cell marker, EBCAM. And so what we do is we direct the cage to one of the tumor cell markers, the key to the other tumor cell marker, and then the healthy cell marker, we direct a decoy which can sequester the key so it can't activate the cage. When we look at the results, we see that we only get cytokine stimuli, we only turn on cytokine production by the T cell, and I'm not showing you this, but then subsequently the killing of the cells by cells which have, actually, I'm sorry, I got that wrong. The 2 tumor cell markers, the print was 2, are EBCAM and HER2, and EGFR is the healthy cell marker in this case. So you can see only the cells that have HER2 and EBCAM are inducing cytokine production. So we're quite excited about this, both for tumor targeting and for specific gene delivery and other places where ultra-precise cell targeting is necessary. I now want to shift gears, and so far everything I've told you about is design of soluble proteins, but I want to tell you about the design of transmembrane proteins now. So Chunfu Zhu designed alpha helical bundles which surround a central pore. This is a design with 6 helices, 2 rings of 6 helices, and this is a design with 2 rings of 8 helices. And this compares crystal structures of the his design models to the actual structures. And after he had verified these formed experimentally, he then converted these into membrane proteins with Pei-Long Lu by resurfacing the outside so that it's now favors interacting with lipids. And when he did this and expressed these proteins in mammalian cells, what he found working with Bill Catterall and Tamara in his lab is that they were quite specific potassium channels. So this is the amount of current in a patch clamp experiment going through the channel. You can see with potassium, there's a much higher signal than sodium. And this is interesting because, as many of you know, there's been discussions for many years about why potassium channels are specific for potassium. And here we made this fairly simple channel. Here's the lining of the channel. You can see here, and this is the radius of the constriction. So this glutamate is where the biggest constriction is. And this fairly simple channel, we already have potassium selectivity. So now it's very exciting. We can start trying to modulate this. We're trying to modulate the selectivity and introduce gating by manipulating the lining of the pore. And just as a demonstration that the ion is actually going through the pore, here's an experiment where Chunfu put a cysteine residue right at the entrance here. The wild type protein is not, the wild type channel is not susceptible to blocking by an alkylating agent. That's the control here. But when you add it to this mutant version, the conductance goes away. And so this is further support that the ions are actually going through the channel. We've also made larger pores, which can transmit molecules much larger than ions, molecules up to about a couple kilodaltons, in fact. And this is from that 8 transmembrane helix ring helix. And you can see the pore in the middle. And I'm not showing you here the experimental data that we can actually measure fluorophores going through this. And in a real breakthrough, Anastasia Vorobyeva recently succeeded in designing proteins which transmembrane beta-barrel proteins. So what I've showed you about so far with the transmembrane proteins were helical ones. But here she's designed all beta sheet proteins shown here, which she purifies in detergent and then adds to lipid detergent or liposomes, and they spontaneously form these structures. You can see there's a comparison of the crystal structure to the design model here. And what was very interesting about this is it turned out the key was really weakening the strand forming propensity of the strands. If they had significant strand forming propensity, they didn't form any cola at all, likely because they formed amyloid. And so this was a case where working out the principles of how to design these proteins was really critical. And we're now using this procedure to design pores with design beta barrels with much larger pores. And we're of course excited about applications for ranging from selective filtration to single molecule sequencing, which, single molecule sequencing has been very exciting using naturally occurring pores. And the fact that we can now design them from scratch, I think means that we should be able to make things that are much more optimal for this purpose. We've also been very interested in designing proteins that can mediate biomineralization. So things like bone, abalone shell, there's a lot of examples of biomineralization in nature. And how can we do this ourselves? So Harley Piles designed proteins that have a perfect lattice match to inorganic crystal lattice, in this case, mica. So you can see here's the mica lattice, and here's this designed regular repeat protein, which has groups which are perfectly aligned with the mica lattice. And when he adds this protein, this is a collaboration with Shui Zhang and Jim DiOrio, it orients on the mica lattice. And Harley was able to go on and then design interfaces between these repeat proteins that direct the formation of these hexagonal lattices. And by changing the number of repeats in the repeat protein, he can make lattices of different dimension. So this is the experimental atomic force microscopy data, and you can see these progressively larger lattices. And so this is really neat. So if you need very, very tiny wells on mica, we know how to make them. And we're now trying to turn this around to drive formation of different types of inorganic crystals by this type of templating approach. I also wanted to mention that we're really big believers in trying to involve the public in science. And so we've developed this game called Fold It, where you start with an extended chain. And in some puzzles, we give people sequences and they have to fold them up. In other puzzles, we just ask them to design proteins freestyle. And they have actually designed many, many very cool proteins. And we've made them in the lab and they actually fold. So it's citizen scientists can now design proteins too. Just to give you a feeling of where we're going, most of the structures I should describe to you were pretty static. Alexis Courbet has been designing rotary systems on the way to rotary motors. So he's designed axles shown in gray and rings shown in blue here. And so this is what the axles look like, and here's what the rings look like. And he's got quite a number of different versions of rings and axles that he structurally characterized. He then adds the axles to the rings, and he can get them to self-assemble, as shown here. The rings, the rotors assemble on the axles. And then he can design the interface here. And this just shows EM pictures of different assemblies he has. And you can see they have quite different symmetries. In some cases, they match D3 with C3. In other cases, they don't match D3 with C5. And those produce different energy landscape, rotational energy landscapes. This is a C3 with C3 axle with C3 ring case. And here the symmetries match. And as a cryo-EM structure solved with Jesse Hansen and Justin Coleman. And you can see that it fits you can see the axle and the rotor. I should say that what we see in the systems is evidence for rotation of the axle of the rotor, because we see much more diffuse density, and that was clear in some of the pictures I showed you. So we're now it's a kind of a tricky cryo-EM problem. Matt just talked about cryo-EM, but you can imagine we have all these different states, rotational states we want to resolve. And of course, beyond that, we would like to be able to drive the rotation of this rotor. And here you see Alexei and Yakov Kipnis have designed a catalytic site at the interface between the axle and the rotor. And this is an active catalyst. And we're now trying to use single molecule experiments to determine how this affects the rotational diffusion of the rotor, of the ring around the axle. And obviously our aim is to design a system like the ATPase where there's unidirectional rotation. So I promised to say something about deep learning. We have now developed methods for hallucinating proteins through deep learning. And I would be happy to say a little bit more about that if there's time. But let me first conclude here. So I think the fact that we can design this wide variety of proteins suggests that we understand the fundamentals of protein folding assembly. And now there's really a huge range of applications. I never dreamed that I would be in the middle of a pandemic using these methods, which seemed totally crazy, just 8 or 9 years ago, to actually try and design, vaccines, diagnostics, and therapeutics. But these methods are really starting to work now. So let me acknowledge the people who did this work. The antiviral design work was done by Longjin Cao, postdoc in the lab working with Brian Coventry and Ina Goreshnik, and we have had really excellent collaborators for the animal and EM experiments. The diagnostic work was done by Alfredo, Andy, and Byung-ha. The vaccine work is that of my colleague Neil King. The antibody nanocages work was done by graduate student Robbie Devine. The self-assembling 2D lattice is a work of Ariel Ben Sasson, postdoc in the lab. The Novo designed membrane proteins, Chun-Fu and Pei Long designed the helical transmembrane channels, and Anastasia designed the transmembrane beta barrel. The design protein logic work was done by Mark and Scott, who are postdocs in the lab, and Jillian, who's a graduate student. Foldit is the work of many people. In my group, the design efforts were really led up by Brian Koepnick. And the rotary motor work is really the brainchild of Alexis Courbet. If there's time, I can say a little bit more about how we are now hallucinating proteins with deep networks, but I'm not sure how we're doing with time. So I'd be happy to take any questions or.
Yeah, David, you have enough questions. You have enough time.
Okay, well, I'll just go through quickly. I'll explain the idea of deep network hallucination. So as you're probably familiar with, a network that's trained for a task like recognizing images of cats on the internet can also then generate ideal images of cats, things that look like cats but are not any actual existing cat. And we've been developing, actually inspired by AlphaFold 2 years ago, we developed structure prediction methods. We were actually able to improve somewhat on what they had done 2 years ago. Nowhere close to what they've done now, but we could then take these models, which were trained on the PDB, and use them to hallucinate brand new proteins. And so here's the basic idea. We take, in structure prediction, you have an amino acid sequence, and then you predict the structure. We go through predicting a distance map between all residues. So to hallucinate structures, we start with random sequences. We predict a distance map, but it will in general be blurry and featureless because that sequence won't correspond to a folded protein. So we can then optimize the sequence, rewarding only the sharpness of the distance map, but not any specific feature of it. And in this way, this just basically shows the picture of this process. So here we start with the distance map, and this is residue by residue. So it's like a contact map just made randomly. We put it through the network and it doesn't really have much features. But after we do this optimization, we get very strong contact patterns between structures. And we can then take these distance maps and generate structures, and here's what they look like. And so when we do this, we generate all kinds of different structures, and this just shows different types of examples. We cover a really broad range of fold space. And we've made a large number of these proteins in the lab, and it's really cool. They actually fold up, and here's their CD spectra. They're very temperature stable, and we've been able to solve structures of a few of them, and they're pretty close to the hallucinated structures. So it's pretty neat. So here's an example of a comparison of a X-ray structure of a design to, or of a hallucinated structure to the hallucinated model by the network. So we can make up these brand new structures. There's also an interesting feature. We can also do another experiment, a calculation where we keep the structure fixed and ask the network and use the network to optimize to find a sequence which maximally encodes that structure. And that's kind of interesting because in what I've told you about so far with Rosetta, I told you we take a target structure and we try and find a sequence with very low energy in that structure. But it's always possible that they're off-target structures that appear. And so we always have to check afterwards to see if that sequence actually folds to this structure where there's lower energy structures. When we're doing the optimization that I just described, we instead can really optimize for this structure being the lowest energy structure because we're trying to maximize the probability of the sequence encoding the structure. And then the final thing we can do is we can sort of combine these two. We can specify part of the structure, but not all of it. So we can just specify the part of a structure that forms some functional site and then hallucinate the rest of it. So we're trying to say, trying to come up with proteins that can be very strongly predicted with the network that have a functional site intact, but where we're agnostic to the remainder of the protein. And I'll just give you some examples of cases where we can take now native structures, take functional bits, and then hallucinate the rest of the protein. And we get quite different solutions. Here's a mimic of insulin, for example. Here's a mimic of a all beta sheet protein. Here's a mimic of the IL-10 cytokine. In all cases, this is the native complex here. And here are little proteins we've made that recapitulate that binding surface. And finally, I'll just mention that another thing that we've been working that turns out to be very relevant for design is accuracy prediction methods. And we found, this is work of now Hiranuma, we found that these deep learning accuracy methods can be very impactful for protein design. So Ivan really developed TR Rosetta, and with Sergei Ovchinnikov did a lot of the sort of foundational work to allow this hallucination to take place. Sam and Temuka characterized the proteins. Chris and Basil really investigated this idea of designing whole free energy landscapes. And Doug and Sydney and Ju have been working on this scaffolding of binding sites using the method. And yeah, and then, well, sorry for that extra bit, but that's, now I'm very happy to take questions. And I guess I should go to Brella, is that right? And I followed the instructions for muting. Let's see.
David?
Yes.
You don't mute. You have to answer questions. Let me read the questions for you.
Yeah, if I can just stay, can I just stay on Zoom? Yeah, yeah. If you can just read the questions, that would be easy.
Yeah. So the question is that David, do your larger cages have sufficient cooperative assembly features to avoid having a range of intermediates, or are there optimal sizes for limiting potential heterogeneity?
That's a really good question. For all of the assemblies I talked about, building cooperativity in is very important. And because if you're, as the question suggests, if they aren't cooperative, you would get a whole distribution of partially formed states. But in fact, they are cooperative because they fold back on themselves. And so in fact, we get very homogeneous populations. We really don't see any sign of partially assembled states because the closed state can make so many more interactions with itself.
Okay. Actually, I have a related question, David. So have you been able to design a allosteric protein, for example?
Yes, well, it's interesting. So the sensor I described basically couples luciferase activity to the binding of a target, for example, the spike protein or a small molecule. It's not quite the strict definition. So in a sense, it's allosteric because its action at a distance is regulating enzyme activity, but it's not really in the traditional sense where you would have a small molecule binding somewhere and causing a local conformational change, or you know, like a change in orientation between subunits. So we're currently working on that sort of system now.
Yeah, I'm sure you are. So another question, hi David, is the LCB1 peptide stable in acid/base as well as in high temperature?
Well, it will be stable under conditions where the polypeptide chain is stable. So, you know, if you go to very high pH or very low pH, you'll start breaking the chain chemically. That's probably below pH 3 and above pH 10. But within that range, it will be very stable.
I see. The next question is, your lab has lead the way to solve protein folding problems. What about the RNA folding problems? How far are we before AI will be able to accurately predict RNA tertiary structure?
Yeah, I think a problem for deep learning approaches is that there aren't as many RNA structures. You know, you need a fair number of examples to predict structures. My former postdoc Riju Das, who's now at Stanford, has been developing methods using Rosetta for predicting RNA structure and designing RNA structures.
Yeah, I guess you're right, because the AI has not overcome that problem. That it's a method you need to learn from somewhere, right?
Yeah.
If there is nothing you to learn, then AI cannot do nothing.
That's right.
Okay,
that's right.
The next question is, the hallucinating protein concept is beyond excellent. Not that the rest is not exceptional, but the hallucinating protein is extraordinary. Okay, that's a praise. It's not a question.
Yeah, okay. Well, thank you.
The next paragraph is, do you envision that automated prediction method will play essential roles in other structural biology approaches? Are there any resources for predicting binding partners for unstructured short motifs, parentheses so-called SLIMS, which are thought to contribute to many short-lived interactions?
Let's see. So there are two questions there. First of all, I think that these deep learning prediction methods will have actually we with our structure prediction methods, which are obviously not as good as the ones that DeepMind just released, we have found that with cryo-EM maps, if we can have a relative, this is work collaboration with my colleague Frank DeMaio at the UW, if we have a relatively low resolution cryo-EM map, we did this recently with the Fanconi anemia complex, where the majority of the subunits in that complex do not have known structure. We can predict the structures quite accurately with our deep learning plus Rosetta approach, and then it's very easy to dock them in the map. So we were able to take a number of density maps which were just not interpretable and very rapidly build models of them. And again, my colleague Frank DeMaio is really focused on them. And now with these improvements from AlphaFold, I think those sorts of methods will become even more impactful. As far as predicting what peptides a protein binds to, that's the long-standing predicting function from structure problem that structural genomics faced. And I think, again, you could imagine deep learning approaches to that, but you would need a fair number of examples. But I think basically you can think about structure prediction giving you as much of an insight into function or close to that as what you would get by solving a structure experimentally. Obviously, if you had a structure of the peptide bound, that would tell you more than the free structure.
Yeah, so now more praise, David, from Gabriel Ludenko says, David, we are blown away. I guess many of us do. And from Catherine Skin, wonderful work with ACE2 receptor mimic. And also, okay, from Catherine again, can you talk whatever about how deep learning affects your work?
Yeah, well, so you can see in that last part of my talk, all of those designs, all of that was being done with deep learning. And I also mentioned that this accuracy prediction network is really guiding what we're doing. So deep learning is having a big effect. Obviously, now we are rapidly trying to incorporate the latest insights from DeepMind into what we're doing. So it's having a very big impact, and for me it's very exciting. I think the things that we'll be able to do will really increase rapidly now.
Great, David. Any more questions from the audience that you can type in Umbrella?
I think at this point, Jianping, probably we should move on.
Okay.
That was a fantastic work, fantastic talk, and to the extent that David has time and anybody else wants to type something in the chat, and you can figure out how to get to Brillo, you can engage with some of the other participants there. So what we'll do now...
I'll just thank everybody for their questions. And
thank you.
Thank
you for both speakers.
And thank you so much. Yeah, we appreciate it. Great work. So now let's move on to the two trainee speakers. The trainees will have 10-minute slots, which means 7 minutes for talking with 3 minutes for questions. So if necessary, I'll interrupt just to let you know, the speakers know that they're nearing the end of their time for talking. So there's some time for responding to questions. The first speaker is a postdoc, Vinicius Contesoto, who got a bachelor's degree in biological physics and a PhD in molecular biophysics from Sao Paulo University in Brazil. He's a postdoc with Jose Unerchuk in the CTBP at Rice University, and he's going to talk about exploring the energy landscape of chromosomes, transitions between interphase and mitotic phase. Go ahead.
Hello, thanks, Professor Jim, for the introduction. And I also would like to thank the organizers for the opportunity to present this ongoing work, which is a collaboration between Rice University and São Paulo State University, where in this work we are trying to do some modeling and try to understand how the chromosomes change shapes during the cell cycle, going from the interphase to the mitotic phase. So if we consider that 95% of the living time of the cell, it spends on the interphase, where the cell passes through different checkpoints, and in this growing phase, it's prepared itself to the cell division and have another cell, right? And then a lot of things start to happen when the cell reach a point to the cell division and go to the mitotic phase. Particular, what we are interested in that the chromosomes change the shape, so the conformations change along this whole process. And then, as you can imagine, it's quite complex. There is more than 4,000 different proteins associated to this process, where we know that condensates and top isomerase play a very important role on doing such, on helping those transitions, right? So for doing our modeling, we will be based on these very nice high-C experiments from Job Decker Lab, where they were able to have those contact maps in a very synchronized cells during these different time points. So we can kind of follow how the chromosomes change shapes from the end of the interphase to the pro-metaphase, and also see these contact maps, how the shapes, the conformation of the chromosomes are changed in these 2D maps. There is a lot of interesting things that we could appreciate from those maps, but I just wanted to point out, if you are able to see that during the cell going to the pro-metaphase, the cell cycle, the chromosomes, they lose what we call the compartments, and we start to observe the appearance of a second band, a second diagonal here. So this is kind of reflect in the polymerase scaling, which it is this contact probability as a function of the dynamic distance, where I will present some results based on this map on the interface and the pro-metaphase, where they have two very distinguished polymerase scaling, which is kind of related to how compact your chromosome will be, right? The model that we are going to choose, it's called Micron, which is this minimal chromatin model, where it's just based a homopolymer potential with bonds, angle, torsions, a very standard one, plus two other terms that we call first ideal chromosome, which is shaded blue, which is a potential who acts along the polymerase chain. It's a kind of a lengthwise compaction term. And the other one is shaded orange here. It's what we call chromatin types, which cross through the space, which is basically the energy interaction between beads of the same or different colors, right? So this potential is based on maximum entropy. And what we needed to do, it is a minimization process where we need to obtain the optimal parameters of gamma and alpha so they can match the experiments. So we do the simulation, and then we compare the results with the experiments and see how far they are from each other, and then we minimize and converge those parameters until we have the simulations agreeing with the experiments, right? So I will just jump into the results that, as we expected, once we have our training parameters, we are able to reproduce the experimental high-C maps. So the simulations on the bottom, the experiments are on the top. So we will obtain the same phase separation. And consequently, we also obtain the same polymerase scale, which means that our simulation is kind of represented well the degree of compaction of a chromosome in their interface, which is its first result. And this is one of the representative structure that we can have in the interface, where the coloring here, it's based on the sequential index, which goes from blue to red, which is head to the tail. We did the same analysis, the same procedure for the chromosome in the pro-metaphase, where we also are able to reproduce the high-C maps, these contact maps, especially by losing the compartments when you are far from the diagonal, and the appearance of this, the second band, the second diagonal, which is reflects on this little bump here on the polymerase scaling. And this is one of the representative structures that we can obtain from this modeling, right? So if we look a little bit closer, we see that there's some kind of helical order here. And the idea is that we are trying to quantify those helix and the elasticity of them, right? So for that, we calculate a orientation order parameter, which will try to go and see those helix formation along the whole polymerase chain. And then the kind of signal that will be obtained looks like a kind of a radio signal, where you have all those oscillations, both for the interface and for the pro-metaphase. Those oscillations means that there is this helix formation, right? Particular for the pro-metaphase, besides those small oscillations here, what we observe, there's this kind of a big wave. If you are able to see here, follow my my pointer. It is we are kind of interested to see what is happening with this big wave. For that, we perform a Fourier transform in this sequential space, where in average you observe a 220 kilobases per turn in the interface for this helix, and for the pro-meta phase, we saw in average 250 kilobase per turn, which means that in the mitotic phase, the chromosome is a little bit more condensed, and those helices is a bit more thinner, right? And in the Fourier signal, we also observe this really intense peak here that in average correspond to 4 megabase per turn. So if we coarsen our structure in this 4 megabase, what we observe are those kind of this major big helix inside the structure of the mitotic, which is what the experimentalists observed in microscopy, what they call these different layers. In a sense, our mitotic chromosome is formed by a helix of a helix in this kind of structure, where the green is the polymer that we are actually simulating, and red is the backbone of this major helix that we also observe.
About 1 minute, 1 minute
Okay.
left. Sorry.
So I just wanted to mention that all the tools and then to these structures that we have are available at the Nucleome data bank that we are hosting here at RISE. The same as for the computational tools that we developed the Open Micro, so we can run the simulations using a single or a multi-chain running in GPUs, which is quite a very good performance. And also on this in the MDB that hosts both experimental and simulated structures, you can visualize those structures there. You can browse, you can overlay epigenetic tracks on top of it, and I invited you to take a look there. And then with that, I just wanted to acknowledge all the collaborators, especially Professor José Orochik, that is hosting me at CPDP, and also the postdoc Antonio here from São Paulo and also from CPDP. With that, I would like to thank you for your attention. Obrigado.
Obrigado. So thank you very much. Interesting talk. We have a couple minutes for questions. One question I have is, how computationally intensive are these simulations? What kind of computer time do you need for, some relevant, some time scale length or something like that, or length scale?
So it is a very good question related to the time scale. So in terms of the computer, of course, it's computationally expensive, but about the time scale of the experiment itself, we don't have this time window frame, because we consider that all high-C maps will be done in equilibrium. So the time for us, it's something that we are not taking account of these dynamics. But from some of our prediction, it's those transitions happen in order of minutes, but the location of each bit happens in order of microseconds.
And how much computer time do you need to simulate on the order of microseconds, roughly?
Yeah, so running the GPUs is a kind of between 24 to 30 hours of simulation to have those high-C maps running. Using CPUs, this can go for weeks. Yeah. Okay,
well, thank you so much. I'm sure others participating might send you messages through the chat or individually. Very nice talk. Let's get ready for the next speaker. And thank you very much. Next speaker is Jim Zong. So make sure you get yourself ready there. So Jim is an undergraduate student who's actually in an accelerated BS, MA, PhD program. So he's somewhere along in that process. So I'm not exactly sure what to call him, graduate, undergraduate, some kind of hybrid. He's works at Rice University in Jane Tao's lab, and he's going to talk about cryo-EM reconstruction of a covalently linked viral fiber. Go ahead, Jim. I see your presentation.
Okay, cool. Thank you, Dr. Briggs, for that introduction, and to, of course, the organizers and hosts for all your hard work. It's good to be here. Right, so today I'll be discussing some of my work on the structural analysis of a covalently linked viral fiber with single particle cryo-EM. So exactly what viral fiber I'm talking about here, this is ORSE. It's the only known virus capable of naturally infecting C. elegans, which, as many of you probably already know, is a pretty handy model organism for biological study. It's incredibly well characterized in terms of the genome, anatomy, and overall development. So ORSE's discovery provides a particularly exciting potential to sort of poke at the pathways underlying eukaryotic virus-host interaction. That being said, there's still much about this virus that remains to be characterized. And if we really want to capitalize on the opportunity that ORSE presents, studying the virus itself, of course, goes hand in hand with that. So what do we know? Well, ORSE overall is a relatively simple, compact RNA virus that contains two segments of positive sense single-stranded RNA. The first strand encodes for an RNA-dependent polymerase, which comes into play during genomic replication, while the second encodes for the structural proteins that are ultimately incorporated into the viral particle. First, we have the capsid protein monomer, which forms the icosahedral protein shell that is the viral capsid. And then we have delta, which is particularly interesting since it can either be covalently fused to the viral particle to jut out from the capsid surface, sort of like I've cartooned here, or simply expressed as a free-floating protein. All of these elements ultimately come into play at different points throughout ORSE's replicative cycle, much of which remains to be characterized. First, it's strongly suggested that the fused form of the delta spike or fiber plays a role in enabling host attachment and entry. These fiber and receptor interactions ultimately enable uptake and uncoding, releasing the RNA genome, which can then be directly translated and amplified to allow the viral life cycle to continue. But you can see here there's a particular gap in our understanding, or in our current understanding of ORSE viral entry and encoding, and much of that can be attributed to the fact that we're not really sure what the infectious virion looks like. Both the capsid and truncated form of the free-floating fiber have been previously solved recombinantly, but there can be a lot of key information missing from these models, namely, ultimately, how the two proteins come together in the functional infectious virion and in exactly what ratios they come together. So with that, we set out to devise a method to culture and purify this virus directly from the C. elegans host, which together with our lab's previous crystal structures should serve as a sound candidate for single particle cryo-EM modeling. Our amplification scheme overall had us infecting large-scale batches of C. elegans that were grown in liquid media, which was then pelleted to collect the culture supernatant. That supernatant was heavily concentrated before finally being suspended on top of an iodixonal gradient for density-based protein purification, leading to this distinct virion-containing band. This method resulted in clean, pure fractions of isolated ORSE virions, which underwent the standard cryo-EM workflow and particle classification, which, although wasn't necessarily required due to the symmetry of the viral particle, served as an efficient means to immediately toss out any faulty, low-resolution particles, slightly reducing our overall data set and refining it. In the end, we were left with this preliminary 2.9 angstrom model, which you can see here really clearly depicts both the capsid surface and interior, depending on what contour you're looking at, and is exciting in that it tells us the delta fiber is distributed along the capsid's five-fold axis of symmetry, proving a long-standing hypothesis based on the multimeric state of the protein. But other than that, you can see that there's quite a bit of information missing on the viral fiber itself. They were essentially reduced and smeared to knobs. And as for why, that can be attributed to the fact that our initial modeling was solely based on the internal icosahedral symmetry of the viral capsid. This sped things up computationally, but it failed to consider the fact that this fiber might be asymmetrically distributed, resulting in sort of the smearing effect. So to compensate, with our next round, we made sure to align particles solely relative to the fiber itself. And in order to do this, we essentially boxed out a region about the viral capsid to include only the surrounding five-fold axes perimeter, where the fiber and its containing vertex would be. So this essentially allowed our initial data set to be reprocessed and classified based on subparticles, where every individual virion particle contributed 12 additional subparticles that could easily be classified as either fiber-containing or fiber-empty. All of this, practically speaking, really allowed us to just zoom in on the particle and recondense the electron density that had previously been scattered. And here is sort of the effects of that subtraction with that boxing in, boxing out. This yielded a much more comprehensive view of the fiber and the surrounding capsid region. You can see here a quick before and after on the left, of course, the initial model solely with icosahedral averaging and the knobs. And then following this boxing in subparticle classification, we get a much more refined focus structure that reveals the full length density of the fiber. And this really improved upon resolution. For example, you can see here, following this subparticle classification, you can really make out the N-terminal alpha helical domain, followed by what we recently term as the beta bracelet motifs that comprise the intermediate regions, and overall providing us with a look towards both the fiber and the penton that contains it. One area that we're actually particularly interested in is this surrounding capsid region, because here we want to be especially on the lookout for any unique features about this asymmetric fiber-containing vertex that might provide hints or clues towards mechanisms for entry, uncoding, and genome release. We're also curious about this particularly small but never before seen 29 amino acid linker region that ultimately is responsible for covalently bridging the capsid and the fiber, of which this model provides our first ever look towards. Ultimately, this method of vertex-based particle alignment will really allow us to work through our next rounds of data collection and particle analysis, which will be especially critical to refining resolution to a point where we can fully map out both the vertex and the linker region. This initial data set is only based on initial screening, but we're really excited to see what about this particular spike protein and region of the viral capsid ultimately enables host interaction, entry, and infection. And with that, I'd like to thank the members of the TAL laboratory for their support and guidance, Junhan Pan, our collaborator, responsible for these incredible cryo-EM models, and of course, those in the audience for their time. I'll be happy to take any questions.
Well, thank you so much. Very, very nice talk. I don't see any typed questions in the chat right now. And since we're running on a little bit on the late side, I hate to do this to you, but I think we should probably move on to the poster session.
Sure.
Again, very nice work. I really appreciate you giving the talk for this session. I wanted to thank all of the speakers for this morning. I think you've heard really a series of really excellent talks, going from sort of molecular simulations, therapeutic discovery, molecular structure, prediction, folding, design, and to, again, sort of data analysis and structure fitting. So I think it's been hopefully it's given those of you in the audience who are more on the experimental side of things, a better context for understanding what kind of things are reasonable to ask of a computational person, and maybe ideas of collaborations that that you might propose. And so let me again thank all the speakers and remind you that we do have a poster session that should have started a few minutes ago. And I'll ask Dawn to share her screen and show us how to get over to that poster session and remind us. And then I think we may have a word or two from possibly from John Tanger as a kind of a wrap up for this morning. So Dawn.
Okay, so please join our posters. Very easy to do. On the left side of your screen, it says poster presenters, click on it. And you'll see the list of our 11 posters. You click on the title of any of them. You can view the poster, find out more about the presenter, their abstract. Feel free to chat with our presenters here. And you'll also be able to see the presentation in a Zoom room that will open for each poster. And our poster presenters will be in their Zoom room for the next 40 minutes. Thank you.
Yeah, thank you, Jim. I didn't really have much to add, except I really appreciate the people typing in their questions to chat. And please remember that the purpose of bringing these speakers and presenters together is to give everyone a chance to talk to them and interact with them on their work. And I hope you'll now do that in the posters. Thank you.